We'll start with diffusion today and discuss decay, secretion, and absorption because eventually we want to build the avascular tumor simulation, and that considers secretion and absorption as a key ingredient of that model. And it's very common in biological simulations to use these techniques, diffusion, decay, and secretion, because cells do that all the time. So we'll get familiar with these concepts before we start building our models. Just quick tips before the lecture. You can store information in cells, individual cells, by using our cell dictionary capability. So you can store all kinds of information. It can be word, it can be a value of float, a list, a matrix, a NumPy array. It could be a torch tensor. It could be a model, another model inside each cell. So you can use cell dictionaries to handle all of that. The syntax is very straightforward. You get your cell object cell, then dot dict, and then in square brackets, this is basically a Python dictionary. Inside the square brackets, you give it, inside quotes, the name of the key, and then equals, and then you assign a value to the corresponding key. You can do that for steppables as well. So if you have multiple steppables and you need to share information between them, there is also a steppables dictionary, which works in a similar way. All right, so the learning objectives of today's lecture, of this first lecture, is to understand the basic concepts of diffusion and get familiar with the mathematical concepts. So there will be a little bit of math or a little bit of showing math and not doing math. And that's important because these concepts we will revisit and use to understand what's going on in the simulation. So it's a little bit like when you are learning how to use a microscope and you learn about Snell law and other equations and you derivate them. They they are useful because if something happens with your microscope, you have a better chance of understanding and handling it, and you understand better what you're seeing and why you're seeing things. And it's a similar way with our models. We see our simulation running and it's doing something, and if we don't understand why it's behaving a particular way, these mathematical concepts help us understand that and therefore help us tune the simulation parameters to achieve the behavior we expect. So, and the topics will be what are fields, how we represent fields, types of fields, diffusion in biology, how the environment affects diffusion. Then we'll visit the diffusion equation. We'll talk a little bit about boundary conditions and what are the meaning of different boundary conditions. And then we'll give interpretations to the diffusion coefficient. So diffusion is critical in biology because signals get transported in time so cells can communicate with each other quite far away. Sometimes these signals can get in ducts and vessels and be carried away in a fluid and reach other cells. And they can, for example, one of the areas that I study in infection, they can prepare cells further away from infection to the arrival of the pathogen. So it's a means of communication, short to mid-range communication and long-range communication if ducts are or blood vessels are playing a role too. By the end of the day, we'll have explored a few examples using diffusion, secretion, and absorption. One is a bacteria ecosystem, and we'll see cool patterns emerging from that. And the avascular tumor-spheroid, A very simple simulation where we have nutrients and they diffuse in the cells. If the cell don't get enough nutrient, it dies, and then the tumor is self-limiting. So what are fields? Fields are physical quantities assigned to each point of space and each moment of time. So as you move in space, you might see a different value to this physical quantity. And as you move in time, you might also see a different value of that quantity. So you could imagine that as, for example, the density of something across space, the temperature in your room, and as opposed to, for example, something that is not dependent on space and time, for example, um, the GDP of a country. I mean, it's dependent on time, but not dependent on space. A scalar field is an example of a field. It's basically the variable type. If you come from a computer background, it's one value, one real value associated to each point in space and time. For example, temperature. We represent these scalar fields using heat maps, using surfaces, isosurfaces. So isosurfaces are contours or surfaces uh, where in that line, the isoline, or on the surface, the isosurface, the value of the field is the same, it's constant. You might see that in maps where you are, for example, checking the elevation of the map. A single line represents an elevation value. In the case of a heat map, a certain color represents a certain value of the scalar field. And in the case of a shape like this, a curve, the height of the curve represents the value of the field. There are also, it doesn't play automatically, okay. There are also vector fields. So for example, in flow, velocity of a fluid, that's a vector field. So it's basically something that has a magnitude and a direction for each point of space and each point of time. In biology, you could have fluid flow, but also fiber orientation, cell polarity, and others. So a couple of questions for you. Give an example of a scalar field, a vector field, and another type of field that you think are important in biology. Is chemical a scalar field? Is electrical? What type of field is electrical? Does a binary field count as a scalar field? Yes. What type of binary field are you thinking about? So for scalar fields, you could have temperature, concentration. For vector fields, you could have polarity, fiber orientation, force. And for other types of fields, you could have tensor fields like stress and strain. Yeah, that's a perfect example of a binary field, whether a point in space is occupied by a cell or not. Yeah. It could be whether the cell at that space is alive or not. That can also be a binary field, whether it's infected or not, whether it is cell type 1, cell type 2, cell type 3. That's also, that would be an integer, right? An integer field, a discrete field. Because the focus here is on diffusion, we will stick to scalar fields, and a very particular case of a scalar field, the concentration field, the concentration of a molecule. In biology, it can be, for example, cytokines, oxygen, glucose, and others, growth factors, hormones, etc. These molecules, they tend to spread, and this is what diffusion models, is this tendency of these molecules to spread around. Of course, that's not true everywhere all the time. So sometimes there are pumps that are forcing the molecule out of somewhere or into somewhere, but um, it's one extra mechanism on top of diffusion. It's not because these other mechanisms exist that diffusion stops existing. Uh, and one particularly important concept is the gradient of a field. The gradient of a field in the diffusion equation tells us where the flow of mass is going to. And the gradient is not the same as the field. It is itself a field, another field. It is a vector field, and it points in the direction of increasing concentration or increasing intensity of the field. Okay? So in this example here, we have a shape, we have a surface. Each point of this surface, x and y coordinates, there is an intensity, a height, and for each of these points, there is an associated gradient. The gradient points to where the concentration is increasing, and it is higher in magnitude as higher the slope at that point. So when we say gradient, we mean the derivative of the field, which has a direction and has a magnitude. That's a vector field. And that's different from the scalar field, which just has an intensity. Let's see if we understand what a gradient is. Can you tell me what is the gradient in each of these points? What are the characteristics of the gradient in each of these points of this curve? Option 1, the gradient has positive absolute value and points to the left. Option 2, the gradient has positive absolute value and points to the right. And option 3, the gradient is 0 for each of these 4 points. And these are the answers. Gradient is 0 on the top and on the valley, on the peak and on the valley. Then on point B, we have the gradient pointing to the right in the direction of increasing intensity. And in the point D, we have the gradient pointing to the left. All right. So now let's talk a little bit about diffusion. And the first question I have about diffusion is, what is driving diffusion? What is pushing these molecules or these particles away from each other? Thermal energy, concentration difference, pressure, all of those, force. I'm going to give negative reviews to the comments that that lead to the wrong understanding of what diffusion really is. Okay, so that's reinforcement learning, all right? So current topics in machine learning, but during the lecture here. Gradient difference of what? Gradient of what? All right, so why is pressure, force, particles pushing each other apart, repulsion, forces, and why is that leading to the wrong understanding? Because even if you do not have any force in your model, even if you do not have any pressure, diffusion still takes place. So that's not the underlying reason of why diffusion takes place. Isn't the title the answer? Let me read the title. Diffusion is passive transport of material from high to low. No, that doesn't answer the question. I mean, passive is giving a tip, but it's not giving you what is the underlying reason of why this is taking place. So there is also a comment about Brownian motion, but like let's just take a look at this video here on the right. In this model here, also known as Ehrenfest model, the particles don't have Brownian motion, they do not interact with each other, they are not subjected to any force, they just move in a straight line. That's all they do. But you start with two compartments. The compartment on the left is full of particles, the compartment on the right is empty. And just because of that, just because of the difference in concentration of these two compartments, and the cells are moving in random directions, there is a higher chance that you observe a particle going from the left compartment to the right compartment than from the right compartment to the left compartment, simply because there is a difference in quantity. There is no fluctuation. The system is completely deterministic. There is no thermal fluctuation here. There is thermal energy, but there isn't like Brownian motion. That's what I'm trying to say. So this is I'm not showing you a stochastic process either. This is fully deterministic, and this is diffusion. Um, So Kyle asks, so would entropy capture a deterministic case? The answer is no, but I'm not going to go into detail because it's too deep. Uh, it has to be stochastic if you pick random initial directions, no? Hmm. Um, so a random initial condition, I don't know if I would categorize that as stochastic. Um, But your thinking is right. Like there is something random somewhere. Yeah. All right. So key characteristics of diffusion before we move on. It's passive. It doesn't require um work. Okay. You don't need something pushing something somewhere. It does not require interaction between the particles. It happens spontaneously. It is irreversible. It assumes, oh, I write thermal fluctuation here. I am wrong. It assumes there is some kind of thermal energy in the system. And the flow of matter is proportional to the difference in concentration of two points in space. Okay, that's a Fick's law. That's how you derive diffusion equation. There is also this video over here. Lovely video. Imagine this could be like a temperature field. This is now representing the concentration of these molecules in space. There is a concept of force that you can derive from this, but it's not a Newtonian force. It is a thermodynamic force, something different. Diffusion in biology is critical because it's a cheap way of transporting molecules. So molecules that can transverse the cell membrane, the cell doesn't need to do anything. The molecule will eventually get to the cell and the cell will use it. It doesn't need like microtubules or a blood vessel. Of course, depending on the distance, because diffusion takes place in such short distances, it's not a long range um, process, not a long range transport mechanism. For large tissues, like more than 10 cells deep, the tissue starts relying more and more on active transport, like vascularization and other things. Um, in the case of cells, there can also be active transport, so for example, through membrane, and passive transport through membranes as well. If it's passive, we can model it as diffusion. And diffusion can be altered by the environmental conditions. For example, the molecules can bind to things, and that can dampen diffusion. And things can absorb these molecules and trap these molecules. One example that I am familiar with is, for example, virus spreading on a tissue. Virus in water has a very, very fast diffusion coefficient. It spreads very quickly, but it binds to mucins and binds to cell membranes, transmembrane proteins. So it doesn't diffuse as fast as you think it does because there are so many interactions taking place with structures in the system. Yeah, what is the difference between passive and active means of transport? So active transport needs work. So in biology, it's for example ATP. It needs energy. It needs the cell needs to expend energy to let it happen. So for example, a membrane pump is an active transport case. And passive is when it just depends on the difference in concentration. So for example, imagine oxygen transport through a membrane. That's passive transport because it just depends on the difference in oxygen outside and inside the cell. So mathematically, you can derive this equation using Fick's law, where the transport of matter is proportional to the difference in concentration in space. It becomes if you assume that the diffusion of these particles, the diffusion coefficient is isotropic, it's the same everywhere. You arrive at this equation where the derivative of the concentration in time is equal to the Laplacian or the second derivative of the concentration in space. And the the outcome behavior, let's say, of this equation is that if you start with a bunch of peaks and valleys, they tend to flatten out. So the higher the peak, the faster it flattens. The lower the peak, the faster it flattens. And so basically, this shape tends to smooth out as time goes. And the outcome will also depend on the boundary conditions. So how the field interacts with the borders is critical for the outcome of your concentration field. So for example, you could this is an initial state. You start, for example, with a droplet or something on like a medium, and then it spreads around. And because we are modeling a small piece of a system in our simulation, we have to determine how it interacts with the borders. And there are three main ways we can do that. The first way is by setting a periodic boundary condition. In the periodic boundary condition, the field, when it crosses a boundary, it reappears on the opposite border. So in this case, the field is crossing the lower border and it's appearing on the upper border. And same thing happening here on the left side. It disappears from the left side and reappears on the right. So it's a little bit like the asteroids game where you or the snake game, those are arcade games where you move, you cross the border and you reappear on the other side. The second common boundary condition is a constant derivative equals zero. That means that the boundary does not exchange material with the field. So if it's a constant derivative equal to zero, there is no exchange between the boundary. There is no interaction between the boundary and the field. You can think of that as the wall of a petri dish. And finally, constant derivative equals zero is there is interaction taking place here between the boundary and there is an exchange of material between the boundary and the field because the field is holding the concentration at value zero. So whenever your field reaches the boundary, if it's a positive value, it's going to lose material to the boundary. So this is useful to model, for example, a reservoir, an infinite reservoir that is um, pulling material from the field, pulling material out from the field. All right, so let's exercise these boundary conditions. Oh, no, there is another slide before that. When the diffusion coefficient is spatially heterogeneous, which means it has a different value in each space and time, uh, you have to use directly Fick's law. You cannot use the diffusion equation itself. Um, but CC3D still solves the approximated diffusion equation, so it still considers that diffusion is isotropic. But when you are inside another cell with a different diffusion coefficient inside that cell, it's going to use its own diffusion coefficient. Oh, yeah. Yeah, so that's confusing, but this is just to mention that in the CompuCell3D, we are not solving the equation that I'm showing you now. We are solving the approximated homogeneous diffusion equation. In diffusive signals, you see something spreading, something being transported, but it's not like in a wave where you can track a wavefront or a peak or something like that. Because you see something traveling through space and time, there is an apparent travel time, but it's not like a signal, a wave, where the travel distance is proportional to the travel time. So that's why I'm calling it an apparent travel time, also known as diffusion time, where the typical travel length of your signal depends on the square root of time. It goes with the square root of time. It doesn't go directly with time. So if you wait like 10 seconds and the field has spread about 10 micrometers, if you wait 20 seconds, the answer is not 20 micrometers of travel length. Okay? It goes with the square root of time. I urge you to be very careful when using this kind of concept, such as diffusion time, because it's not intuitive, such as travel time of a wave or like a bullet or something like that. Diffusion time is like less intuitive. It is not very precise, let's say. Um, so be careful with that. There is more description here of like why it makes sense to talk about diffusion time. And if you want to be very strict, you will want to set like a concentration threshold, and then once you measure the apparent travel time of that concentration reaching somewhere in space, you can more accurately talk about the diffusion time. But this you can use, you still can use this to compare with travel times of other signals or forces. For example, if a cell moves at a certain speed and you're trying to see if another signal reaches that point faster in time scale, approximate time scales, you can use diffusion time. And it goes with L squared over D, where D is the diffusion constant, diffusion coefficient. All right. So you can use it to compare timescales, but be very, very careful because it will, if you use it recklessly, thinking it is the right answer, like, oh, this is my diffusion time, so this is the time it takes for my signal to arrive somewhere, you will be wrong. I promise you. So use it carefully and ask us questions if you're trying to use it for your specific problem. So summary of different boundary conditions. Periodic boundary condition, the field crosses a border and appears on the other one. Constant derivative, there is no exchange of material between the field and the boundary. And constant value 0, there is an exchange and the boundary acts reservoir. It could also be another value here. So if it's a constant value 10, it's a reservoir holding that field concentration at value 10. Now let's jump to exercises. So in this exercise, we have different cases where we wish to assign an appropriate boundary condition to. And in this first example, we have an epithelium, or like a slice of the epithelium, a cross section. The top part is the epithelial barrier. The bottom side is where you have like another type of tissue. And on the sides, left and right, you have the same epithelium, the same kind of system. Okay? And we wish to assign a boundary condition for each of these three cases. So there is a boundary on the bottom part, on the upper part, and on the sides. And the question here is, what is the appropriate boundary condition for each of these cases and why? So I'll give you 2 minutes to give me to think about this problem and answer in the chat. Okay, I'm satisfied with the answers in the chat. Um, um, all of you are right. So yes, no flux on the top part because the barrier doesn't allow exchange of this molecule with the outside. If we are having more of the same kind of system to the left and to the right, using periodic boundary condition is the way to go. And on the bottom side, we have another tissue type that could act as a reservoir, for example. It allows exchange of this molecule with the boundary. So it's a matter of getting what is the concentration of my field inside that tissue. And then that's the value you're going to use as your constant value for the third boundary condition. Okay, let's take a look at this other case. We have a gland and it's producing a hormone or like some substance, secreting a substance here. And then we have a cavity, which is this boundary here. We are not modeling the cavity explicitly. It goes in the model in the form of a boundary. And so how does the field interact with this boundary? Is it possible to combine different boundary conditions in a single CC3D simulation? Yes, absolutely. So let's reason a little bit about this. Your cells are secreting constantly. They're always secreting that this is usually how glands work. They are always secreting something nonstop, right? So yeah, let's assume that's the case. If you do not have communication between the gland and the border, such as derivative zero, constant derivative zero, your gland will accumulate uh material until it explodes. So that's not like very natural to me. Constant derivative equals zero. There must be an escape of this molecule being produced by the gland. It has to escape somewhere. It escapes to the cavity, to the boundary. So constant derivative zero can't be. If you set a constant derivative negative, that's even worse because if your gland stops producing something, the field will go negative, which doesn't make sense. Unless you are interpreting negative concentration of hormone and some, I don't know. I don't know. Constant positive derivative. If you do that, then the cavity will always be supplying the gland with material. So I don't think that's a good answer either. Is periodic reasonable? If it's periodic, you basically have another gland above, right? It's like if you have the same kind of thing, but over here you have a gland here, right? Like this. This is the case of periodic boundary condition. So your gland is supplying hormones to another gland, to itself, let's say. It's supplying hormones to itself. Doesn't make sense to me either. So the answer here is constant value, and then you find what is the value that the cavity, the typical concentration value of the cavity. You can adjust that. It can depend on time, but I would put here a constant value greater or equal than zero, some kind of value here. If you have negative flux again, if you have negative flux at the boundary, and the concentration of field is zero inside the gland, your field will go negative, which is not physical for the concentration of a molecule. So this is similar to the third boundary condition in the previous example. Here we have like a reservoir here, and the cavity behaves as a reservoir. It's like you have some sort of smell inside your house, and then you open the door. You open the outside door. The outside works as a reservoir, while the nasty thing you're cooking on the stove is the gland, which is filling the space with this nasty smell of your food, and then you open the back door and it dissipates into the environment, and the constant concentration of that smell in the environment is zero. Last example, we have a tumor and two possible boundary conditions here. The fifth boundary condition is you have ECM continuing forever on the sides, and on the bottom part you have a blood vessel that is supplying oxygen to the tumor. So oxygen is our concentration field in this case. And we're just modeling the tumor and the ECM. We're not modeling the blood vessel. The blood vessel is our boundary. What boundary conditions would you choose for each one of these? Okay, I see a good contrast in the chat. So there are some people believing that boundary 5 is a constant value, and some people believe that boundary 5 should be periodic. Now you are free to fight in the chat, okay? Hmm. Is there no oxygen flow with the ECM? See, if there is no oxygen flow at all in the ECM, the oxygen will never reach the tumor because there is ECM here. There is ECM here everywhere around the tumor. There is ECM. So if the ECM doesn't allow oxygen flow, the oxygen will never reach the tumor. So the ECM is definitely permeable to oxygen. So there is exchange here in the boundary. It cannot be a constant derivative zero. So I'll give you one more minute to think about it because I think that people are thinking a lot about this problem. So I don't want to waste the opportunity of students thinking. Okay? In the era of AI, that's a rare thing. All right, let me give you a reason to why, in my opinion, this here is constant value above zero, something above zero. I feel we ought to impose some constraint on the derivative too. Would expect the blood vessel to deliver a constant flow of oxygen plus be a large reservoir, so constant derivative and value. No, no, no. So the blood vessel does not supply a constant flow of oxygen. Imagine the tissue had more oxygen than the blood vessel. Would the blood vessel still pump oxygen into the tissue? No. The tissue would be supplying the blood vessel with oxygen. Oxygen transport is passive. So you know you don't have pumps on the wall of the blood vessel pumping oxygen into the tissue. It is passive. It is a constant value greater than zero. Now, if we set periodic boundary condition on 5, we are assuming that there is another tumor over here and that there is another tumor over here. Is that what we are modeling? Or are we modeling just one tumor? So a periodic boundary condition, if we are modeling just one tumor, is not appropriate. And if your argument is, oh, but the walls defining the ECM, they are too close together. They're playing a huge role. Yeah, so make them further away. But this is constant value as well, and this is constant value as well. There is and you could make it a constant value here that is different from a constant value over here. It could be a constant value that depends on the coordinate y. So it could be like 100 here and 0 here, and then 50 here in the middle. But if you set periodic, you are modeling an infinite series of tumor side by side. Does it make sense? It does make sense. Um, so the correct answer is constant for both. The ECM could be assumed to be some constant average level, and the blood vessel flow is happening on a far faster time scale, and so can be assumed constant. It's Eddie's answer. And it's also Shamayita answer. Yeah. So constant value in both seem the most appropriate choices here. So these are the answers, and let's move on. In CompuCell, you can select different solvers, different types of solvers. These are the main ones. So you can use one that gets you to the steady state immediately. It is a little bit more expensive because it calculates the field profile until it stabilizes. That's useful if diffusion is very fast and you don't care about the transient. For example, if you have a system that changes in the scale of days and you have signals that saturate and equilibrate in a matter of minutes, using the steady state diffusion solver can be appropriate. The diffusion solver FE, that's our regular one. It is the faster diffusion we have, and it's useful if you care about the transient. What happens when the signal is generated until it stabilizes this process. If this process is relevant in your model, then you use the diffusion solver FE. Finally, we have two reaction diffusion solvers that they are very similar, the diffusion solver FE, but they allow you to couple fields by writing reaction diffusion equations. And in version 4.10, which I think isn't released yet, solvers are GPU compatible, so they will speed up your simulation quite a bit. Now for our final exercise, we're going to open Twedit and import a chemical field to our simulation. So open your wizard, and then you will find this page or something similar to this. This is how you add a chemical field to your simulation. You are going to type a name. Um, there is a typo here, oxygen with a C. Oh, it's with an X here. Okay, it's fine. So oxygen could be the name of the field. Then you select the solver. Here I'm using diffusion solver FE. Then you hit add. It's going to show in the table, and then you can hit next. So I'm going to give you 2 minutes for this one. There are other ways you can include fields in your simulation, and I will show them later. Let me open my Twedit here. So here I'm going CC3D project, new CC3D project, and I'm going to skip straight to the chemical field. And there is the window. And I will type oxygen in the field name. I have diffusion solver FE selected. I'm going to hit enter or click add. It shows on the table right. Let me know if you need more time in the chat. And I'm going to proceed. So in the next step, right here, this other window. So now I'm going to go full screen here in the presentation so we can see better what's going on. We have a bunch of options for setting up our field. For example, you can have multiple fields to start with, and you can change the settings of each of the fields you have in your simulation. So in this case, we just have oxygen, but if we had another field type, we would have another tab here, one tab per field, and then you can switch and change their settings. You can set you have global settings. So if you set diffusion coefficient and decay global, it's going to act everywhere. And then in places where you have a cell, instead of using the global parameters, it's going to use the cell specific parameters. So the cell parameters overwrite the global parameters. They take precedence. And medium, for diffusion and decay, medium uses the global settings, okay? And you cannot set global secretion, global uptake, these kinds of things. You can set a diffusion coefficient for global that influences medium and all cells that you don't have a value. And for each cell type, you have a specific diffusion coefficient. So you can have a molecule diffusing faster on medium and diffusing slower inside cells. You can also set a decay. The decay is how fast the field fades away. So there is a loss, a regular loss of material happening everywhere. And you can set that global, which is going to take place in medium, and also a cell type specific decay. And the other settings are for secretion and uptake, and we are going to visit them later. You can also control secretion and uptake using Python. And in this case, you check this box, and then CC3D will not write anything about secretion in the diffusion servers, and will allow you to set up your own secretion and uptake settings via Python. Why would you do that? Because Python scripting is more flexible than XML and Wizard. So if you want to do something very specific, very creative, using Python code is the way to go. You can define the initial conditions for your field. For example, using a mathematical expression, following the mu parser notation. These are basically C and C++ symbols. And you can also define your field initial condition using a text file. Finally, the boundary conditions, you can choose between constant value, constant derivative, and periodic boundary condition. And you choose them for each axis and each side of the simulation. So if you have a 3D simulation, you have 6 faces. So for the x-axis, you have the left face and the right face, and you can set boundary conditions there. For the y-axis, you have 2 faces, the one below and the one above, and for the z-axis, you have the one the front, the one the back. What is the relationship between the diffusion coefficient in CC3D and the experimental, the real life diffusion coefficient measured in units of meters squared per hour or per second? So to establish this relationship, you have to establish a relationship between pixels and real life distance units. So if you say 1 pixel is equivalent to 1 micron, this is how you establish a relationship between the length of a pixel and the real life distance measurement. And then you also need a relationship between Monte Carlo step and real life time units. Once you have the relationship between time and space, experimental and simulation, then you can convert your diffusion coefficient from real life to diffusion coefficient in CompuCell3D. So you first need to establish a relationship between pixel and distance units, real life distance units, and Monte Carlo step and real life time units. So for this exercise, please set the decays to 0, and then set the initial concentration expression to this expression here. This is basically a droplet at the point x equals 20, y equals 20. You have the initial concentration 100. This is what this expression is doing. So the equals here, this is C and C++ syntax. It's a logic operator. It's asking whether x is equal to 20. If that's true, it returns a Boolean true. And then if y is equal to 20, it returns a Boolean true, which means if both are true simultaneously, the product is true, and then 100 is the final value. 100 times true is 100. If either x or y are different than 20, then it's 0. Okay, because false times 100 is 0. And then you have 0 everywhere except at x equals 20, y equals 20, where you have 100. And then set periodic boundary for the x and y axis. And in this example, we don't have the z axis settings because our simulation is two-dimensional. Yes, and people are already finding the mini game where you have to press OK every time you type something on the initial condition. And this is something you can definitely complain about. Yes. So this wizard page is new. So we are using you as our experiment. Okay. So any issues you find, as James says, please report. And this is one of them. Once you type that, you can also copy and paste. That's another option. And it should work. Well, that's the mini game. Let me run the mini game here myself. So you can copy this, paste it, and there we go. Now here I'm going to set periodic, and that's it. And then we can hit next and finish. And now, yeah, before we run, you can also add a diffusion solver, Steppables, using the Twedit drop-down menu. So that's another way. You don't need to use the wizard. Although the wizard is currently the best way to set up your diffuser field. And once you have them here, you cannot use the wizard again. So that's another problem that I already reported on GitHub. And which is if you come here to CC3DML Steppables and you hit diffusion solver FE and you hit replace, it's not going to open that wizard page for you. It's just going to replace whatever you have with a huge diffusion solver code, but it contains all the options. All the possible options are here. So you would have to manually edit this monster code, even give a name here, the etc. Okay? But that's another way. So hit finish and then find the diffusion solver FE steppables in your XML. So over here, I have on line 38, I have my diffusion solver FE. If you added cell types, it's not line 38, it's going to be larger, greater than line 38. And then you will see your oxygen field with the diffusion constant, the decay constant, the initial concentration expression, the boundary conditions, and you can edit these parameters here on XML. Okay. I already posted on GitHub a request for a feature where you launch that same window again, and then you add it on the window, and then when you press enter, it edits here for you. But currently you have to edit by hand, and the wizard window just allows you to do an initial setup. You can also find your decay settings in the XML, secretion settings. If you set any secretion setting, you can also find them in the XML. Um, the boundary conditions, and the initial concentration expression. Then you can run your simulation and so open in player and run. And here I'm seeing the cell field, but if I come here to oxygen, I can now see the field, and here's my droplet on the corner, spreading, diffusing. Okay. So you should see something like this. Let me know if that's what you see. And depending on the visualization settings you set, you might even see the field spreading through the boundary because you set periodic boundary conditions. So if you let it spread long enough and you have visualization settings to see such a low field value, you will see it crossing the boundary as well. If you specify a boundary condition and an initial condition that happens to be on the boundary, which one takes precedence? They will take place simultaneously. Ali, it seems like you have you don't have the periodic boundary condition. It seems that you have the constant derivative 0. So if you go to your XML and check that, I think you have the constant derivative equals 0 there, which is not a problem, okay? It's part of the homework exercise. If you want to change the way you see the field settings, the field visualization settings, I'm going to show you how to configure the field visualization for the 2D visualization. So if you go to Tools and Configuration, there is a tab called Field, and you select the field name in this drop-down menu. For example, oxygen. If you have multiple field options, you can select which one of them you want to change settings. And then you can change, for example, the minimum value and the maximum value. This is the field range, the magnitude range of the field. And you can also unfix that so it changes automatically. Like as your field spreads, the maximum goes down. So this allows you to see the field spreading like more easily if the field is changing over time everywhere. I don't have a configurations option under tools. Ah, what is this, Mac? Yeah, this is this Mac or Linux? Mac. Click Manage Simulation Settings instead of configuration. Manage Simulation Settings. Or you can also just hit this button here. It's like a panel with sliders. Oh no, oh it's not. Manage Simulation Settings. Oh, it's something else. Do you have this screwdriver and wrench symbol in your player that says configuration, and then it pops up in a window like this in Mac? Oh, so on Mac you click CC3D on top and top left, then preferences. Okay, so that's in Mac. Okay. And also check if you have this screwdriver wrench symbol. That symbol over there. And then here, for example, you can, just as an example, I'm not doing scalar contours, but you can do. If you want scalar contours, you hit show. Um, and I'm going to unfix these two. Yeah, it doesn't let you. Yeah, so Ali is right. So scalar contours don't show, don't allow you to change. Yeah, so that this is a bug, and I'm going to report it in GitHub. But oh no, here they are. If you hit isolines, they at least generate. So these are the isolines, the white contours here. Let me see if after, okay, so I hit show, I press OK, and I reopen the tools, and now I can set the ISO values. That's fine. And there what I did is I unfixed the maximum, and if I keep this running, I can see it spreading and spreading, spreading and spreading, and not like fading away forever. And eventually, because I'm using periodic boundary conditions, I will see the field crossing the boundary and reappearing on the other one, on the opposite side. Let's see. Yeah, so if I go up here, I already see the shade of the field appearing on the opposite border, in the upper border. And if I go to the right border over here, I also see it because I'm using periodic boundary conditions. All right. So that's it. That's it for the lecture. To generate the circular contour type figure, you need one, to remove your decay, to set the diffusion coefficient everywhere. You need to set an initial condition of a droplet using that expression. Also, set up your periodic boundary conditions. Then you can, if you want to visualize it better, you can go to Tools, go to your simulation settings, and then you unfix the maximum value of the field, so it tracks the field as it spreads. I can see a small circle in the bottom left part. Yeah, that's correct. And then you can zoom in and drag your field of view so you can see that droplet spreading. My diffusion doesn't go that far before disappearing. How do I correct that? So there are two options here. One possible option is you have decay. The second possible option is you don't have decay, but you could go to the configurations and unfix the maximum field value. So as the field spreads, you rescale the magnitude of the field down. I can't enter any values in ISO values. Yeah, what Ed is suggesting something what worked for me is you hit OK and then you reopen the configurations. All right, so now I'm going to address a question by Udis. This is probably very outside the scope of this workshop, but if we want peaks to spread out, I would maybe expect dCdt equals d times norm gradient c, not Laplacian. So the diffusion equation, you have dCdt equals the Laplacian of c, okay? The Laplacian of c is the concave. Okay, so if your concave is like this, the Laplacian of c is lower than 0. And if you have a smiley face, the Laplacian of c is greater than 0. If Laplacian of c is lower than 0, it means that over time, your concentration will decrease. So I'm going to draw in red the next point here, the next configuration of the field. So it's going down because you have a sad face. And this one you have smiley face, so it's going to go up like this over time. Is this clear? So this is what the diffusion equation is doing. This is why it makes sense to use Laplacian. Now, what you were suggesting is del C del T equals the gradient of C norm. So let's see what this means. Let's see together what this means. So acceleration would be if you had the second derivative of time. That would be acceleration, right? This. Sorry, there's a 2 missing here. This would be acceleration, right? The Laplacian is a difference of differences. So let's say you have a place here, a site in space, a site here and a site here. And here you have like 3 molecules. Here you have 5 molecules, and here you have 1 molecule. Okay. You have transport from here to here and transport from here to here. So this guy here doesn't change if you have this setup. It's only going to change if this guy here also has 5 molecules. Then you have transport from both directions to inside, and then this guy here will be greater than 0. So it's really the smiley face or the sad face that you are looking at, that you're interested in. So if you just have, this is a difference. So this is dc dx. This is a difference over here. And you have another difference over here, dc dx. But what you are really interested is the difference of the difference. So you are interested in dc dx at point x minus dc dx at point x minus dx. And that's your second derivative. And that finishes my Q&A. Thank you.